So the first thing to point out is just an important concept about introns. And if you think about the actual gene here, you can kind of see the notes. If we have this long gene and you have sections that are introns and sections that are exons. Basically the idea is this, that depending on what the spliceosome treats as a particular intron and exon will change the value. So in other words, if this is treated as the gene, and then let's say this region here now becomes the exon in between those spots, and then this becomes the gene, we now have, so here's the exon 1, and then this would be like exon 2 and exon 3. That is a very different expression than the original coloration that I had for the different, where the reds would have been the exons. And so the idea is simply this. We used to think that there was this idea that there was one gene equaled one polypeptide or protein, but now because of this RNA splicing and because things can be spliced differently and how they are actually spliced, we can get a lot more proteins. The number of proteins that are possible is greater than the number of genes. We used to think if you had 10,000 genes, you could produce 10,000 proteins. Now that is not the case. You have 10,000 genes, you could produce a lot more just depending on how it's spliced. Once again, giving more variation, more diversity in our population. When we got done with transcription, we made this RNA. We had it from our DNA. It was a process of transcription, created a primary transcript. We then edited down the information to our actual mRNA molecule, and that is going to leave the nucleus and go out into the cytoplasm to do the next stage, which is translation. And that's going to be the focus of this video here today. So when we talk about the genetic code, we need to understand that it is a code. And that means usually when you have a code, you have a key, something to crack that code. And so for that reason, the code that you need will be provided to you. It's not something that is memorized or anything like that. You just use it to take the information and convert it into the language of amino acids. And that's what we'll kind of talk about today. You store that if you looked at the activity of the lab in class last time. What's important about this genetic code is we use codons. So we take a sequence of DNA. The first thing we need to do is convert it into an mRNA molecule. And then here, we break this into 3-letter codes, and that's how we go about doing that. And it either corresponds to one of 3 things. The codon will correspond to the start codon, to any of the other amino acids, or it doesn't code for anything, and therefore that is a stop signal or the terminator. The other thing that's interesting with the genetic code is that it is nearly universal, which basically means that whether you're us, a bacteria, whatever, use the same genetic code. You don't have to use a different key for bacteria, a different one for animals or plants or anything like that. And so GUC codes for amino acids in valine, in bacteria, eagles, dogs, your own cells. Now what's really cool, because of that universality of the code, this is where we can actually take codes, insert the DNA into another organism, and get the genes from one organism into another. This is where that term GMOs come from, genetically modified organisms. We'll talk more about that in class and kind of going forward about that. GMOs kind of have this negative stigma, and we'll talk really what a GMO is, why there's some concern for them, and why they're useful as well. Okay. The big reason why this is important, not only can it allow it to go in there and actually go and edit the code, add our own pieces, but it also means that because the code's so universal amongst all organisms, probably came from a common ancestor. Now, one of the things that you see here is that we have 20 different amino acids, and when you talk about the fact that there are 3 spots and 4 letters, 3 codons, 1, 2, 3, and 4 letters, you can think about the number of combinations that there actually is. And what you can actually do, since there's 3 letters and there's 4 different combinations, you can do 4 to the 3rd power. That's the number of different ways that you can create using those 4 letters combinations. So if you think about it using mRNA, you could have A, you could have T or a U, you can have C in any combination. So that's 4 to the third combinations. If you actually calculate that out, so that's 64 different combinations, but there are only 20 amino acids. And what this tells us about the genetic code is that we're going to have some redundancy built in, that there will be more than one code that codes for each amino acid. So for valine, there will be several actual codons, sequences of DNA, sequences of mRNA that'll code for that, okay? And when 2 codon, what typically we see is the variation comes in this 3rd letter. So in other words, when they do code for the same amino acid, the first 2 letters are the same. Using valine as our example, GUU codes for valine, GUC codes for valine, A codes for valine, and then GUG does as well. There's redundancy. Because they all share the first 3 letters are the same between all 3. GU, the difference is in that last letter. We call this idea the wobble hypothesis, that every organism will use the same codons for the amino acids, and any of the variation will happen there, which tells us this. When we talk about mutations, which will be in class on the C day, what matters is going to be that where the mutation occurs. If I take this valine right here, and a mutation occurs, gets hit with UV light, and it changes this to GUC, it'll still code for valine. That mistake's not that important. And so if we have an error at that 3rd letter, we are still going to be okay. Once again, the genetic code builds in some safety checks to prevent mutations from impacting. Where it'll matter is if I have a mistake somewhere in these first 2, typically that's much more detrimental. So if I went from the original GUC to GGC with the change in that 3rd letter, I now get glycine. And so now a very different amino acid than the one we previously had. Okay, so those things matter. Where the mutation occurs matters. Okay. Once again, the fact that we have this idea that the code's the same for all living organisms tells us that there is redundancy here and that it supports the theory of evolution. So here's the typical way you see this and how you read a particular code is you take a sequence of the genetic code, and let me kind of type this out to show you. So here I have a particular sequence of code, and what you want to do and how you take this code. So this is a sequence of mRNA. It's already been converted from DNA to primary transcript. It's been edited to this form. And what you do is you start on the left, and you're going to read left to right, and you find the first sequence that corresponds to the start codon. In this case, it's AUG. And so you don't have to necessarily follow it you don't start the triplet codon until you find it. So you just kind of scan along, and then as soon as you find it, there it is, you break it up into 3s, okay, which I'm going to attempt to do, hopefully with a little better handwriting. Like so. Okay? And then all you do is code. So at this first location, I find AUG, so AUG, that's my start codon. I just write down here MET. Put a bond, because that's going to be bonding it together to the next 3 triplets, which is CCG. So I go C, C, and then I get the 3rd letter down here, which is G. I put pro. So that's C. This first C tells us it has to be in this section. The second C tells us it has to be in this section. So therefore it's in this box. And then we have G, which tells us it's PRO. Okay? Continuing on, I have, so there's my UGA again. Now, since once again, you can't have a start once you already start. You just write MET again, you're fine. Okay, and then I go to CGC, so, CGC is ARG, and then I get to this, U, G, and A, that's our stop codon. and that's it. So this is our sequence of amino acids built off of this. Anything after the stop codon does not matter, okay? That's how you use the genetic code. It does the exact same thing as the image to the left. It's just a preference. On the test, I will always provide you with both of these. With this one, all you do, same concept. First letter is the biggest circle. So if we were doing this code, AUG, A, and then I find the U, and then I find the G, that's our start codon. Okay, so for this one, CCG, I go C, C, G, that's our proline. Okay, that tells us the sequence of amino acids based on that. That's how you use it. So either one you can use on the test, both of them will be provided to you. The process of this happens, again, in the cytoplasm, and what's going to happen is like a soup where there's a bunch of tRNA floating around. The tRNA molecule itself, the reason it kind of calls that is that it's a sequence of RNA, and it's folded into 3 compact loops. It kind of looks like this. This is where the amino acid's going to be attached. Looks kind of like an upside-down T. Down here's a specific region that we're going to need to know. It's called the anticodon, and it corresponds to a sequence of amino acids. So if I have a sequence that's AUG and my mRNA strand, on the anticodon is going to be the opposite. So A goes with U, A, C. That's what the anticodon corresponds to. What it's going to do is it makes these partner up or match up so we know the next letter to go. Okay, it's complementary to one of the 64 codes. So we talk about this tRNA molecule. There's all these tRNA. They're all complementary. The only exception are the 3 terminators, or the 3 stop codons, do not have a corresponding tRNA. Therefore, when a stop codon comes up in the sequence, there's no tRNA to attach. It's done. The sequence stops, and the amino acid sequence has been built. All right, so you can actually look at the tRNA molecule here. Important thing. This 3' end up here, that's going to be where the amino acid's going to be attached. Attaches corresponding to whatever our anticodon is down here. So depending on what that sequence is, it'll be the anticodon there. So I use the example on the previous of MET for our start codon. The mRNA is going to have its AUG, so we're going to have AUAC would be here in those 3 letters, and in doing so attach the amino acid, MET. I mentioned this in the previous slide, but just important to note, these are the 3 stop codons. Now, it's not saying you have to memorize these, but it's not a bad idea. It just makes your life a lot easier, so you can kind of look for them along with the start codon. But these 3, they do not have a complementary anticodon, and so basically as you're building this amino acid sequence, and you're building these sequence together, it's building like links. And all of a sudden you don't have anything to link onto here because you hit the sequence of code, let's say AUG, and therefore since you have no attachment, this basically falls off, and that's our polypeptide sequence. Okay. All of this is happening at the ribosome, and a ribosome is where the proteins are going to be made. The ribosome has 2 units, a larger subunit and a smaller subunit, typically drawn like that. The mRNA molecule will be fed through it, like so, and then you end up having 3 locations that are mattering, the E, P, and A sites. And each one of those plays a role. Basically, there's all these tRNA molecules all floating around out here, and they're going to move to the A site, which is like an on deck, and then eventually move to the P site, where that's where the position is going to be attached. And then once the tRNA gets to this spot, it's already lost its amino acid, and it'll go back into the collection, be adding another one. So those are the 3 subunits, or the 2 subunits of the molecule for the ribosome. And they only come together, that's an important note, only during translation. So a ribosome is only functioning when it's actually translating the protein. So here's just a definition that'll help you with the 3 sites. A is holding the next amino acid in line. P is the position, and it's going to be actually where the specific amino acid is going to be attached to the growing chain. The E is not mentioned here, and that's where the tRNA molecule, after it's completed its task, exits the molecule. This process occurs when the mRNA bonds to the smaller subunit in the cytoplasm. So here is my smaller subunit. Remember, this is always in the cytoplasm. My mRNA molecule will start to attach. In doing so, when that occurs, my larger subunit will attach over the top, like so. Okay. This is actually our ribosome. So when it's all three parts, that is a functioning ribosome. Okay. And when this occurs, you have your A site right there. Here's your E. When the start codon gets to this location here, that's when we're ready to actually start adding in the tRNA molecules and the process of translation occurs. After the mRNA bounds to the ribosome, they'll start attaching based on the codon. So just transfer those on with each individual particular codon. The tRNA molecules themselves can actually be reused, attach new amino acids, and then be put back in to attach to the growing chain. And so it allows for the fact that if we have 300 amino acids being added, we don't need 300 tRNA molecules. They can be reused. So this basically kind of writes down and shows the sequence of this occurring. You can see the DNA, transcription, translation. There's the codon that goes with it. This diagram, the letters are a little bit off because the sheet's off, but it does at least provide you with some imagery of the process. And remember, this is for one particular gene in the sequence of DNA. Now, when we talk about this for eukaryotes, we mentioned already, there's those introns and exons. The exons are actually the coded regions, the introns are the non-coded regions. And we need the exons are the areas that are actually expressed, and so they'll be spliced together, put together to make a particular gene or sequence. All right, so there's the cutting, the process transcripted piece that'll be actually used in translation. These are two images that I like that kind of basically show this process occurring. Both of them, or excuse me, the one on the left does not have the parts labeled, the EPA. If you want to do that now, you can kind of see this would be the exit site. Where that's at is the P and this is the A. They are labeled over here. And so as the mRNA molecule is being fed through, this one is leaving. This is our next growing chain. It's going to go here. As it moves from this location over one spot, it'll attach the next amino acid to the growing chain. Then it'll be moving the P spot. A new A will come in, and then this one will exit. In this case, that one will move. And so that's how this process occurs, with the polypeptide kind of just hanging out and dangling out the side. When it's all completed, this molecule here, they'll hit a stop codon. When that does, there's no attachment, and the process is complete. So here's basically your homework that I asked you to do, this worksheet about practice. All you're going to do is in the top part up here, you write the process. So what's the process going from DNA to mRNA? That's transcription. I'm going to run out of space. And the process here from mRNA to the anticodon is translation. Okay? And then what you're going to do is go through and actually figure out what the parts are. So CCC goes with the mRNA GGG, which will correspond to the anticodon CCC. And I use this column here to figure out the code. GGG, G, G, and G codes right down there for glycine. I'll write glycine right in the box. Now take a look at this one. T, A, T. T goes with A goes with U, A. Now this is why this is important. Don't write TAT. That's not going to be the anticodon, because remember, tRNA molecules can't have any T, so it would actually be U, A, U. And what does AUA code for? Well, A, U, and then A, right there, is isoleucine, right? So I'd write that there. Run out of space, isoleucine, okay? You'll complete that for all the different parts, but it's a way to practice understanding the code. It's important for you to do that, an easy skill that I think should be done on the test going forward. Okay, so that's all I got. If you have any questions, please don't hesitate to ask. I'm sorry it took so late to go to post this because of a lot with Things going on at the hospital with my dad. Don't worry, he's doing much better. But I appreciate any of you guys who sent me some emails. That was nice. So have a good snow day. Watch this if you have any questions, and then I'll make another video a little bit about what we're going to do because of the missed school. So have a good day. Bye.